Required Practical 10, all about the rate of decay. Why does milk, eventually go sour in the fridge? It's the perfect everyday example of the very process we're going to investigate today. Section 1. Let's solve this mystery of the sour milk and connect it to the big biological idea we're studying, decay. So what is decay, really? Well, you can think of it as nature's cleanup crew. It's just the process where tiny little things, microorganisms like bacteria, break down all the complicated stuff, like the fats in milk, into much simpler bits. And our job in this practical is to find a way to measure how fast that happens. And that brings us to the official aim of this whole practical. Our mission is to investigate how temperature, that's the thing we're going to change, affects the speed, or the rate, of milk decay. And the clever way we're going to track this is by measuring changes in its pH. Okay, let's get into the nitty-gritty, our investigation plan. Here's all the gear we'll need. Now, you might be thinking, doesn't milk take days to go off? And you'd be right. That's a bit too long for a lesson. So to speed the whole thing up, we use an enzyme called lipase. It basically does the same job as the bacteria, breaking down fat, but way faster. We've also got sodium carbonate to make our starting solution alkaline, and this clever stuff called cresol red, which is an indicator that's going to change color for us. More on that in a minute. All right, the method. First up, you're going to set up your water baths at different temperatures. You'll have one tube with your milk mix in it and another with just the lipase. Now, this is super important. Pop both tubes into the water bath for a few minutes to let them get to the right temperature. Then it's go time. You add the lipase to the milk, slam that start button on the stopwatch, and watch it like a hawk. You are timing exactly how long it takes to go from purple all the way to yellow. And as you're collecting that data, you absolutely need a clear, organized table, just like this one. Being neat and systematic is what science is all about. For every temperature you test, you'll write down the time it took for that color change to happen. What is actually going on inside that test tube? At the very heart of this reaction is something called an enzyme. The easiest way to think of enzymes is as tiny biological helpers. They're catalysts, which is just a fancy word meaning they speed up chemical reactions without getting used up themselves. So how do they work? Well, we use this idea called the lock and key theory. Imagine the enzyme is a lock with a very specifically shaped keyhole. That keyhole is called the active site, and only one specific molecule, which we call the substrate, in our case a fat molecule, can fit into it, just like a key. This perfect fit is why enzymes are such specialists. And the specific enzyme we're using is lipase. Its one and only job is to lock onto lipids, which is just the scientific name for fats. And when that lipase grabs hold of the fat in the milk, it breaks it down into two new things, fatty acids and glycerol. And I want you to really remember that first name, fatty acids. And this is where it all comes together. It's brilliant. We start off with an alkaline solution, so our cresol red indicator is a nice purple color. Then we add our lipase. It goes to work breaking down the fats and producing, you guessed it, fatty acids. As more and more acids are made, the pH of the whole solution drops. It becomes acidic. And that's the trigger that makes our indicator switch from purple to yellow. That color change is our finish line. Time isn't a rate. So how do we actually turn these numbers into a measure of speed? Look, what we measured was time. But think about it. A really long time means a super slow reaction, right? Like a snail. A really short time means a super fast reaction, like a rocket. To turn our time measurement into a proper rate, we use this simple formula. Rate equals 1 divided by the time. It just flips it so that a short time gives a big rate and a long time gives a small rate. Easy as that. Okay, thinking caps on. If we actually plotted a graph of the rate of the reaction against the temperature, what shape do you think it would make? Have a quick think. It should look something like this classic hill or peak shape. Here's the story behind it. At low temperatures, everything's a bit sluggish. The enzyme and the fat molecules don't have much energy, so they don't collide very often. So a slow rate. As we turn up the heat, they get more kinetic energy. They start whizzing around and bumping into each other more frequently, so the rate shoots up. That is, until we hit this perfect sweet spot, the optimum temperature. But if we keep going and get it too hot, the enzyme itself starts to fall apart. Its special active site changes shape permanently. And when that happens, we say it has denatured and the rate of reaction just crashes. Let's focus on what those examiners really want to see. First up, variables. I can almost guarantee you'll get a question about these. The independent variable is the one you change on purpose, in this case, temperature. The dependent variable is the one you measure, which was the time for the color to change. And your control variables are everything else you have to keep exactly the same to make it a fair test, like the volumes and concentrations of all your chemicals. Look, no experiment is perfect. A classic source of error here is that everyone might judge the color change slightly differently. It's a bit subjective. A great improvement would be to use a pH meter to get an actual number. Another problem is that a simple beaker of hot water cools down. The best way to improve that would be to use a proper thermostatically controlled water bath that keeps the temperature spot on. Now listen up. If you get a big, juicy 6-mark question asking you to explain your results, this slide is pure gold. This sentence is basically the perfect answer. You have to tell the story. Start with kinetic energy and collisions. Then you must use the term optimum temperature. And finally, you have to explain what happens when it gets too hot. The active site changes shape and the enzyme becomes denatured. Nail those key points in that order and you're flying. Okay, let's boil all of that down. What are the absolute must-remember facts you need to take away from all this? We've seen that temperature is a huge deal when it comes to controlling decay. It's the whole reason we have fridges, right? So my final question to you is this. What else could we control? Think about all the ways we stop food from going off. We salt it to remove water, we pickle it to change the pH, we can it to remove oxygen. This practical isn't just about milk in a tube. It's the key to understanding the science of food preservation. Well done for getting through it.